So yesterday I posted an assignment over DNA and haven't made a video for it yet, so I'm going to make that for you this morning, and so you can use it to take your notes for it and to hopefully help you with that worksheet. So with DNA, the first thing I wanted to talk about was not this. First thing I wanted to talk to you about was an experiment. And you don't have to put this in your note. You can just listen to this. So back in the 30s and 40s, scientists didn't really know what carried your genetic information. So a couple of scientists by the name of Hershey and Chase came up with this experiment. They thought it was either proteins or DNA that carried your genetic material, and they actually thought it was probably proteins, just because there's 20 amino acids that make up all your proteins, where there's only 4 bases that make up your DNA. So what they did real quick, they grew a batch of phages. Now, bacteriophages are these little viruses that infect bacteria, and they kind of look like that. You can Google one and look at it, and they're pretty creepy looking little things, kind of alien-like. But they grew these phages. They grew some with radioactive sulfur, knowing that sulfur was a pretty common component of a lot of proteins. And so then after they grew these, the phages incorporated that radioactive sulfur, which had a dye in it, and they incorporated it into their bodies. And viruses, the only part of their little bodies that have protein in it is their outer coat. So their outer coat would be kind of this pinkish color you can see in this image. They went ahead and injected their DNA into the bacteria. Then they put all of it in a blender and basically just mixed it all together. And then they put it in a centrifuge, and if you don't know what that is, that's a device that you can put test tubes in that spins them around really fast and causes any of the larger particles to go to the bottom. And bacteria are quite a bit larger than viruses, so when they put them through the centrifuge, they noticed that the pellet at the bottom, that it would contain all of the bacterial cell and components. And they noticed that there was none of the dye that from the protein. So that told them that the viruses did not inject their protein into the cell. They went ahead and ran the experiment again, but this time they used radioactive phosphorus, knowing that it's found in DNA and not in protein. Did the same thing, but this time you'll notice that the phages incorporated that phosphorus in their DNA. So they injected it into the cell, put it in the blender, ran it through the centrifuge, and this time the pellet contained the pellet that contained that radioactive DNA from the phage. So a cool little experiment that showed that DNA carried the genetic material. All right, so let's talk about DNA real quick. DNA, of course, we're familiar with the phosphate and the sugars. They make up the backbone of the DNA molecule. Hopefully you notice in this picture here that they don't run like this. They actually kind of run like this. They run opposite of each other. And the reason for that, well, that gives them what we call as anti-parallel structure. So you notice this side, the little oxygen is on top, and on this side the oxygen is on bottom. They run opposite of each other. And this is called the 5' end and the 3' end. And the reason we call it the 5' end is because each little angle here in the sugar molecule, there's a carbon. And so this is carbon 1, carbon 2, carbon 3, carbon 4, and then in H2C there's carbon number 5. So that's why it's on top here, so it's the 5' end. And down here you have 1, 2, 3. The third carbon right here is on the bottom, so that's why this is the 3' end. And it's just the opposite here. You have 1, 2, 3, 3' end, 1, 2, 3, 4, 5, and this is the 5' end. So they run opposite of each other. Okay? So anytime if you have a DNA sequence and it starts off with 5, so if it would say like 5TGCA3, the opposite of that would be 3ACGT5. The other thing I wanted to point out here on these two images, notice how the thymine and the cytosine are single ring structures, whereas adenine and guanine are double ring structures. Scientists, when they looked at the images of DNA, they saw this very symmetrical, three-dimensional, double helix, and it told them that they had to have a single ring bonded with a double ring, because if they would have had two double rings bonded together, it would have been too wide in some places, and if they had two single rings bonded together, it would have been too narrow. But having the double and the single had that consistent shape all the way through it. And then they could actually go in and count how many thymines there are, and that always equaled to however many adenines there were. And how many guanines there were, and that always equaled how many cytosines. So that let them know how those letters bonded together. By the way, you will need to know, I asked the question, if I were given, if you knew that there was, in a strand of DNA, there were 40% thymines, how many cytosines would you have? So the question there, and how you figure that out, hopefully I'll give you a second if you want to try to figure it out. Feel free to pause it. But if you have 40% thymines, that means you have 40% adenine. That leaves you with 20% left. So that would mean that cytosine and guanine would both have 10%. So anyway, there will be a test question on that, and just want to make sure you knew how to do that. All right, let's move on here. So this here is a phosphate and sugar. Remember, they make up the sides of the DNA ladder, and the bases make up the rungs. All the information, the phosphate and the sugar just give it support, and all of the information is in those bases, like adenine, thymine, cytosine, and guanine. And so remember, all adenine always equals thymine, and guanine always equals cytosine. So that's what bonds with each other. This is a strand of DNA that kind of shows you how replication takes place. And I want you to notice here that you have both, that the original strand will separate out, and both of the old strands act as a template to form the new strand. That's called a semi-conservative, and how they form their new strand. So then the two new strands that are formed, the two new double helices, will have one old strand and one new strand. And they will be identical to each other and identical to that original DNA, excuse me. Next thing I want to talk about, remember DNA, before a cell can divide, each chromosome must make a copy of itself. As we talked about DNA replication when we talked about mitosis, remember it occurs during the S phase of interphase, and that's when the DNA synthesis takes place, DNA replication. Without DNA replication, new cells would have half the DNA as their parents, and we don't want that to happen. I'm going to skip the videos for now. You feel free to go in and watch. There's lots of videos out there that show you DNA replication. I would encourage you to go and check some of those out. This here, real quick, I want to spend the rest of the time on talking about this. I'm going to kind of break this down. This will help you on that worksheet. So there are three main enzymes that I feel like that are involved in the process of DNA replication. There are others, but there's three main ones that I want you to be familiar with. The first one is helicase. And helicase is an enzyme that actually unzips or unwinds that DNA strand. Okay, and then that forms this replication fork here. It's kind of like this where it's being spread apart. Okay. And then the second enzyme that plays an important role is this DNA polymerase. DNA polymerase, it says it adds the new nucleotides to that old strand. But the interesting thing about DNA polymerase is that it can only add new pieces of new nucleotides in that 5-3 direction of that new growing strand. So as that replication fork opens up, the DNA polymerase will continue to add new nucleotides going toward that replication fork, and you have this one long continuous strand of DNA of that new strand that's formed. However, but because it can only add in that 5-3 direction, on this bottom strand, so we call this one the leading strand, on this bottom strand here, it won't work that way. This is what we call the lagging strand. So again, because it can only add in the 5-3 direction, this is the 3' end of that new strand. So that DNA polymerase has to start off wherever it opened up and start adding nucleotides in this direction. And then as it opens up more, it has to back up and add more DNA up to that point, and then back up and add more up to that point, and more up to this point. And what that causes is these little fragments that are called Okazaki fragments. And so it kind of works in the opposite direction. So you hopefully can see how we call this the lagging strand. Eventually, another enzyme, the third one that I really want you to know, a DNA ligase, comes in and kind of glues the sugars and phosphates together here and here and here. Here and here, and then eventually here to here, and eventually here to here. So that's how that process works of DNA replication. Helicase unwinds it, DNA polymerase adds the new nucleotides in that 5 to 3 direction. 5 to 3 here too. You have the leading strand, which has one continuous strand, and the lagging strand, which has these short segments that are called Okazaki fragments that are eventually glued together by DNA ligase. Okay, so that is it pretty much on DNA replication. So hopefully this will help you to complete that worksheet.